In this video, we will explain the functions of a few biochemical machines, as well as the relationships between them. The first machine we will talk about in the video is the ribosome, which includes the smaller and more specific mitoribosome. The ribosome is responsible for reading mRNA from DNA and translating that genetic code into proteins. These proteins are then sent all over the cell and can function in other machines, enzymes, organelles, and even structural components like the membrane. The mitoribosomes are particularly important because they code for proteins used in very important processes that occur specifically in the mitochondria. In the mitochondria, which is an organelle in the cell referred to as the powerhouse, there is an electron transport chain and a molecule called ATP synthase. Together, the electron transport chain and the ATP synthase create molecules of ATP, which our bodies use as their main source of energy. The electron transport chain shuttles electrons through 4 different protein complexes, and as each electron is passed through, some hydrogen molecules are moved from the matrix of the mitochondria into the inner membrane space of the mitochondria. When enough hydrogen accumulates in the inner membrane space, a gradient is created, and the natural tendency is for the hydrogen to flow back into the matrix. The hydrogen moves back into the matrix by flowing through ATP synthase. As the hydrogen flows through ATP synthase, conformational changes move the machine such that it combines ADP and free-flowing phosphate molecules into ATP. This ATP is then used for energy in many different ways throughout our bodies. Another way our bodies can create ATP is through glycolysis and the citric acid cycle. The only catch is that some of these pathways, like glycolysis, need to use ATP in the beginning stages before they can make ATP in the final stages. If we look at the pathway of glycolysis, we see that glucose, which is a molecule we consume when we eat, is broken down into 2 molecules of pyruvate. In this process, we consume 2 molecules of ATP, but we produce 4 molecules of ATP. So some simple math shows that the net production is 2 ATP molecules. This isn't very much, and alone it wouldn't be enough to sustain our bodies. So to produce more ATP, our bodies send the products of glycolysis, the pyruvate molecules, into another pathway called the citric acid cycle. However, the pyruvate itself is not what enters the citric acid cycle. It is first transformed into acetyl coenzyme A. This is where the PDC comes in, fully known as the pyruvate dehydrogenase complex. The PDC takes pyruvate, and using a cofactor called NAD+, and a molecule of coenzyme A, it creates acetyl coenzyme A and carbon dioxide. The acetyl coenzyme A is able to enter the citric acid cycle and produce ATP, GTP, and reduce cofactors NADH and FADH2. These cofactors, while not usable themselves as energy, are the molecules that supply electrons to the electron transport chain in the mitochondria. If we remember correctly, electrons moving through the electron transport chain are what contribute to the hydrogen gradient that moves ATP synthase and creates the most ATP of any pathway in our bodies. With this video, we hope you've learned a little about how these biochemical machines function and how they work together to create the energy our bodies need.